Good morning, ladies and gentlemen. It's my pleasure to give you a talk about our research work. The title of my topic is Patient-Specific Induced Pluripotent Stem Cells as a model for studying fatty acid hydroxylase-associated neurodegeneration. In this talking, I will first give you a general information about FAHN, especially the difference between patients and the mouse model, and also describe the strategy for building IPS model and isogenic lines. Then go through with the generation of FAHN specific IPS models and isogenic lines. At last, discuss using IPS models to study mechanism of FAHN. As you know, FAHN is an autosomal recessive disease caused by mutation of FA2H gene. This disease is associated with nucleodystrophy, SBG35, NBIA. The neuron degeneration is brain iron accumulation. The clinical characteristics are complicated depending on the different type of mutations. In general, the age of onset in most patients is less than 5 years old. And they have corticospinal tract problems, for example, spasticity, and a mixed movement disorder like ataxia dystonia. And some patients also have eye problem. Late in the disease course, the patients can have intellectual impairment and seizures. The MRI findings are useful for diagnosis. They contain, for example, white matter change, hyperintensity of globus pallidus, pontocerebellar atrophy, thin corpus callosum, and iron accumulation. FAHN is a subtype of NBIA. Some FA2H mutation, for example, these two types of mutation, can cause abnormal iron metabolism, resulting in iron accumulation in the brain, especially in globus pallidus and the substantia nigra. The reason of the iron accumulation in the brain is still not so clear. Some data suggest that the change of intracellular ceramide pore composition, for example, hydroxy fatty acid containing ceramide and non-containing ceramide, the ratio between them is one reason. And also impaired palmitoylation of transferrin receptor is another one. Still, iron accumulation is a cause of neurodegeneration or a consequence of neurodegeneration is still not so clear. Animal disease model is a powerful method to study human disease. Until now, 2 FA2H mutation mouse model has been generated. The first one is null mouse model, in which FA2H is deleted in all cells, and another one, conditional mouse model, in which FA2H is deleted only in oligodendrocytes. Compared to mouse models, the CNS phenotypes between them are similar. For example, demyelination, Purkinje cell loss, cerebellar dysfunction can be found in both animal models. However, learning memory defects happens only in null mouse model, but not in conditional mouse model. suggests that oligodendrocytes are not responsible for behavior defects. Although mouse models show manifestation of patients, but there are some difference between them. For example, the age of onset, as I mentioned before, usually is less than 5 years. However, demyelination happens at aging mouse at 12 months of age. Furthermore, at early stage in the mouse model, no obvious defect in neurons or oligodendrocytes and astrocytes were found. Therefore, it is necessary to build iPS-based FAHN disease for the study of the mechanism of FAHN. Because until now, the pathophysiology of central nervous system seen as in FAHN is still not so clear. Therefore, patient-specific iPS are excellent alternative to study precise function of FA2H and the mechanism of FAHN. Thank our patients and families for the support. We have already got 2 different types of mutations. One is here, another one is here. This fibroblast, we will use them to generate iPS. But keep in mind, there is still a limitation of this patient-specific iPS approach. The main problem is lacking isogenic control because of difference of the genetic background between the patient and the healthy control. And this difference between them will affect the accuracy of the study. Therefore, it is also necessary to build FAHN isogenic iPS lines. We establish isogenic lines from healthy donors using CRISPR-Cas9 technology to create the non-FAHN disease mutation, for example, like this one. Because this type of mutation is associated with nucleodystrophy, SPG35, and NBIA. Therefore, combine both patient-specific disease and isogenic iPS will help us understand the mechanism of FAHN. This figure shows iPS-based disease modeling. The fibroblasts from patients are cultured by in vitro system, and then they will be reprogrammed by transduction with transcription factors and drive them develop into patient-specific iPS. After characterized of this iPS, then they will be differentiated into disease-affected cell model. In our case, like neurons and oligodendrocytes. As a disease modeling, then the pathophysiological difference between patient and the control cells will be detected. In this case, then we can get ideas about the function of mutant gene and the mechanism of the disease. Also, this iPS system can be used as a drug screening by new drug development for the treatment, and also can be used in regenerative medicine. This panel shows you the generation process of iPS. But still, I should mention here that iPS generation is a routine work in our lab. we have already got the FAHN IPS cloning, but by some reason I cannot show you the pictures. Instead, I show you the Niemann-Pick type C1 IPS pictures published from our literature. So go back to the generation process of IPS. The human fibroblasts are cultivated by in vitro system. This is the cells. And then reprogram with transduction by SOX2, OCT4, and so on, and drive them to develop into IPS. And then they form IPS colonies, just like this. After characterization of this IPS, for example, pluripotency, descent, karyotype, mutation type, and the differentiation potentials, then they will be drive to develop into disease-affected cell types, for example, like here. This figure shows you the assessment of pluripotency in IPS. Immunostaining is performed to identify whether IPS clonings express pluripotent markers. This figure shows exactly that the IPS clonings can express LALOG, OCD4, and SSEA4 pluripotent markers. And also, the descent, karyotype, and mutation in IPS should be also proved, as I mentioned before. The descent of IPS is proved to identify whether genetic fingerprint of IPS is the same to the fibroblasts to exclude contamination with surround cells. And also, the karyotype of IPS should be proved to exclude chromosome aberrations occurring during reprogramming. And the mutation type of IPS, of course, should be also identified by sequence to ensure that conservation of this mutation is same to the fibroblast. And also, the differentiation potential in IPS should be also proved. After differentiation, immunostaining is performed to identify whether IPS can differentiate into distinct type of cells. In these figures, the IPS can exactly show that they express nesting, the marker of ectoderm, and also muscle actin for the mesoderm, and alpha fetoprotein for endoderm. The last part of our research is using IPS model to study mechanism of FAHN. After IPS differentiate into desired disease cell types, the difference between the patient's and the control IPS derived cells will be checked in order to find out the precise function of FA2H and the molecular pathophysiology of FAHN. In our case, we drive this FAHN IPS into differentiate into neurons, oligodendrocytes, and astrocytes. Then in neurons, we will compare the difference between the FAHN patient's neurons and the control neurons. And we focus in neuronal proliferation, migration and differentiation, and also neuronal structure, for example, the axons and synapse, and also some structure. Also the functional property and the synaptic transmission. In this case, when we compare the difference between patient's neurons and control neurons, then we can get ideas about the problem of this disease and get exactly the mechanism of this disease. Of course, same for the oligodendrocytes. We will focus on the difference between oligodendrocytes proliferation, differentiation, and myelin formation. Because in FAHN, the myelin formation problem or demyelination is a bigger problem in both patients and animal models. Also, for the astrocyte, we will focus on the morphology and the activity of these cells and try to find out the difference between the patient's astrocyte and healthy control astrocyte. In this case, get ideas about the disease problem. Because the FA2H is involved in the synthesis of 2-hydroxy fatty acid synthesis. In this case, the membrane lipid metabolism and the lipid buffers in different type of cells, for example, neurons, should be also investigated and try to find out the difference between them and get ideas about the mechanism of the disease. We hope these findings of this experiment will help us understand the disease, the precise function of FA2H, and the molecular pathophysiology of FAHN. Unfortunately, we still haven't started this part, although we have already got the cloning. Because of COVID-19 pandemic in last year, and there is some delay of our project. And I'm sorry about that. We hope that we can report the findings of this data in detail next year. At last, I would like to thank our patients and families for the support and the donation of the cells. And also thanks my colleague, Dr. Andrea Hillman, Dr. Maurice Frisch, and our PhD student, Fatima Effendig. And also thank our cooperation partner, Dr. Venkateswaran from University of Ottawa. And also thanks LIBA NBIA Disorder Association for the financial support. And also thanks for your attention.